Pretty good. We have 11 so far.
Okay. We'll get started in just a minute. Are you going to start the recording?
I just did.
It has not given me the warning message yet, so I was curious.
Nope, just check recording in progress.
Okay, very good. And you say we get going. All right. Well, thank you all for joining today. And my name is Charles Cywin. I'm the Director of Small Molecule Neurotherapeutics Development here at NINDS. And I'm going to be telling you today about the NIH Blueprint Neurotherapeutics Network. To get my thing in the right spot, I guess. And there we go. So the program's vision is to combine the strengths of NIH and industry to create sort of a virtual pharma model around your ideas. You as the NIH investigator bring novel drug targets and novel disease assays and models and a chemical starting point for moving forward into the program. We can provide you the industry expertise in terms of advisors with extensive pharma experience, and we can also provide industry standard contract services. The goal in all cases is to de-risk the potential therapeutics to the point where industry will invest in them, allowing the new drugs to reach patients efficiently. We try to identify the best ideas for translation in the NIH community through our funding opportunities, and the associated infrastructure allows them to move forward. This funding is non-dilutive, which is great for people who are spinning out programs because it makes the program more attractive to future licensors. All the resources that we have available are what you need in order to get to an IND in the small molecule space. And what's really unique about the program is we really strive to preserve the intellectual property to your institution to facilitate that licensing down the road. The program itself is for both academics and small businesses. The grants support small molecule drug discovery and development projects. You can enter either at the discovery phase or the development phase, and the way we sort of view those two things, or if you're still doing optimization of medicinal chemistry, you're still in discovery. If you're really picked your candidate and you need to characterize it further, you're at the development phase. Although some discovery activities do tend to happen even on those kind of projects. So if you do want to do a development phase project, come talk to us. They're for multiple reasons. And I'll get into some of those as we go through the talk today. Opportunity to access the NIH-funded consultants, contract resources, and our expertise internally in terms of project managers and science officers. Our project managers all have experience in moving projects to IND. We work closely to facilitate your team's efforts. We don't get involved in the weeds of trying to do inventive things, but we are there to help facilitate the whole process all the way through. We coordinate all the contract and grant resources and consultant resources that are needed. We also have science officers who have the disease portfolio that your project comes under in their under their purview, and that allows you to get information from them on what sort of the state of the art is from a portfolio's perspective on that disease. We're offering this infrastructure in the network by first developing a lead discovery, lead development team. That meets every 2 weeks. As I said, it's sort of a virtual pharma, so it's just like if you're in pharma, you're meeting every 2 weeks to discuss the medicinal chemistry efforts, anything to do with assay development, PK, drug manufacturing, toxicology, regulatory, all depends on where you are in the spectrum. Any or all of these could be under discussion at a single time. As I mentioned, the grant budget has to include the disease biology. So the primary secondary assay to run SAR, the animal models for the disease, and that effort has to be as part of the grant budget. And then everything else is optional in terms of being in your grant budget. You could have your own medicinal chemistry effort and not utilize our contracts, or you can utilize all of our contracts, for instance. And we have contracts for medicinal chemistry with Curia for PK and tox, and this is both GLP and non-GLP tox. We had drug manufacturing formulation with Curia, MRI Global, and clinical trials with PPD and Duke. And if you're doing optimization, you will use our relational database system with CDD, and that allows us to have a uniform system of looking at data across your project and comparing it if internally, well, not with you, but internally across all projects to make sure things are performing well. And sometimes we do find things in one project team that relates to other project teams in terms of assay trends or something that that actually influence. And I can think back to at one point where we had an anatomy study where we were doing stuff with mass spec detection in one program and with UV in another program, and really decided the mass detection was working much more reliably. And we switched everybody to mass detection going based on what that effort. So you really have to ask yourself the question, do you have the expertise to do this in-house? And if so, do you want to do it in-house or not? If you already have a pre-established relationship with some CRO, you should talk to us about it because we may be able to maintain that. There are budget rules and things that tend to lead you to utilize our contracts, but we work with you to make sure we are trying to be fair about this. The contracts are tailor-made. As I mentioned, they're not dilutive. We are training to retain intellectual property, so the MedChem and DMFP contracts have special clauses in them to make sure they sign the IP to you. The industry consultants have signed IP agreements with you or institutions so that all the IP stays with your program. Progression is always milestone driven, and so it is very much like a pharma model where if things aren't progressing, things will stop. There are 9 institutes and centers that are signed on to the program. They're listed here with their contacts. They're also in the funding opportunity announcements at the back, along with their mission statements on what they're willing to take under the program. As I mentioned, in all cases, the desire is to get to the clinical trial. So that means you're going to write your budget from wherever you're starting all the way to IND. If you're using our contract, then you are not a clinical trial grant. If you're planning on doing the clinical trial under your own budget, then you are a clinical trial grant. But in all cases, we're trying to get to the clinic. Everybody enters what's called the preparatory phase. That first phase is either a UG3 or U44 phase 1. The execution phase, as we refer to it, is the UH3 phase and the U44 phase 2. The UG3 phase can only be 1 year. The U44 can be up to 2 years because that allows you to get up to 5 years in both of these. But most cases, this has a pretty limited budget, so most people under the U44 are still going to probably go for the 4-year option. You can only receive 1 UG3 award, so we're not going to give you a UG3 award and then extend it for another year. It's going to be making a milestone decision to go forward or not. Total number of years of MedChem that we would typically support is 3 years, including the first year of exploratory work, which is less effort than it is in hit to lead and lead optimization. So depending on where you're entering, that's sort of the maximum number of years you can determine that we're going to probably be funding chemistry efforts on it. Everybody, as I said, enters that preparatory phase along the pathway. But as I mentioned, talk to me about doing development projects, because not all of the institutes and centers take development projects. NIDA and NIMH particularly have excluded those projects from their using BPN. Additionally, we talked about those resources. The language in, for instance, NIDA's grant mission statement says you will utilize the BPN resources to the maximum possible. So they're not going to want to see contracts within your grant budget. So again, talk to us beforehand. It's much better to work these things out in advance than afterwards. As I mentioned, there are different budget rules for the SBIR versus the non-SBIR. The UG3, UH3, if you're going to go over 500,000 in direct costs in any year of this grant, and you have to give me all years as I mentioned, both the UG3 and the UH3, up to 5 years of a budget. If any of those years are more than 500K in direct costs, you need to ask permission to come in, which means you're going to have to give me specific aims, a budget, a detailed budget for all 5 years with a justification for why you need to go over that. For the SBIR, that rule doesn't apply, but it does have rules on its own on the budget, and these are in total dollar costs. The phase one can be up to 500K in NINDS specific rules, and 700K across 2 years if you happen to do 2 years, and up to 1.5 million in total costs, up to 3 million across the maximum of 3 years. These are, as I said, NINDS rules. NIDA, for instance, has different rules which have lower budget caps. And so again, depending on what indication you're coming and what IC you're going to, you need to talk to us about your budget if you're going to be coming into the SBIR to make sure everything is the way it should be. If you're applying as a foreign organization, it is okay to do so under the UG3, UH3 mechanism, not under the SBIR. The foreign organization can come in, but the reviewers will need to assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, resources, populations, or environmental conditions that exist in the other countries and are either not readily available in the U.S. or augment existing U.S. resources. You need to make the case for that in your application. Alternatively, if you're working with a U.S. collaborator, the U.S. collaborator can be the institute of record, and you can be a foreign component. The hurdle for that is lower. As I mentioned, all projects are milestone driven. As such, when you come into the program, we will give you sort of detailed milestones, but they go along these lines that you see here. If you're coming in the exploratory phase and you want to know my terminology, exploratory phase is about show me the assays functioning. Do you have Z prime score, that you have good throughput there, that you can reproducibly get results. You have your screening funnel worked out. You may not have all the assays developed yet, but you have a plan in place and know what you want to do there. You have a target product profile, so we know whether we're going after, at least from the onset, an oral drug, a parenteral, or what. And from a chemistry perspective, we can resynthesize the molecule, still get activity, demonstrate SAR is not binary. If you have structural alerts, see whether we can get rid of the structural alerts and still maintain activity and that kind of things. That can all be part of that exploratory phase. We usually do things like in vitro metabolism at that stage. We may do in vivo PK just to get a benchmarking on the best looking compound and see where we are at. When we get to hit to lead, our focus is really about pharmacology, showing in vivo activity by any route of administration, chemistry, trying to get from maybe multiple series down to the series that we want to look at in lead optimization. Once we get to lead optimization, it's about picking the candidate from that one lead series, establishing our exposure activity relationship, our PK/PD, and then moving on and trying to do an acceptable rodent dose range finding tox to show a therapeutic index. Once you pass that stage, you really are at the development stage and you picked a compound. We will then go into things like large animal dose range finding studies, determining what stable form we want to do, a salt screen, a polymorph work, etc. We have to make sure we have a tractable synthetic route that we can actually produce the material for toxicology on scale enough to enable phase one. It doesn't need to be the commercial route. It just needs to be economical enough that we can produce the material within the budget of BPN. You need to onboard your clinical specialist to help design the clinical trials that are coming down the road so that we make sure we're running the toxicology necessary to do that work. IND enabling is all regulatory effort. We'll have to have completed in the end lead optimization, all the in vitro parameters that we needed to do, and then we start doing the in vivo tox, safety pharmacology, genotox, repeat dosing, and large animal and rodent, etc. We'll then move on to opening the IND, and typically most people run the single ascending dose through our contract work in healthy human subjects, although you can propose other things. As I mentioned, we try to protect your intellectual property at all times. Inventorship is always determined by U.S. patent law. There's no way around that. BPN itself is not seeking any stake in your IP just because you're in the BPN program. So prior to the award, we're going to ask that you internally have all of your stuff worked out with your collaborators. So if you have multiple institutions working together, are going to want to know that the IP agreement is in place between all your institutions so that there won't be any argument at the end on who's going to prosecute the IP, who's going to file the international patents, and all that good stuff. They should aim for unencumbered intellectual property, however. The goal is to create a licensable product. We have a wealth of information available on our website, and I encourage you to go to the Blueprint Neuroscience website, and you will find BPN there. And BPN not only is here, but you'll also find at the bottom a blueprint for BPN for biologics, which I'm not going to talk about today, but there's also a blueprint for devices under MedTech as well. If you don't have all that preliminary data we talked about in the beginning, there is a program NINDS has for its mission, and some other institutes have R21s and other things to do similar type of work, that allows you to get the data you need. So Ignite program is a great program to run, develop a screening assay or an assay and then test compounds in them, and to find that starting point. You can also have developed the therapeutic and run in vivo efficacy studies, or develop an animal model if you don't have it yet, under these 3 funding opportunities. They have regular receipt dates. The next couple are in February and June. Rebecca Roof is the contact, but the goal of this program is to get you the preliminary data to enter BPN or BPN Bio type programs. Again, it's an R grant, not a U grant, so they're not cooperative agreements, but they are still milestone driven, so you will have to give a definitive milestone that allows you to make that transition from R61 to R33. Their maximum of 750,000, maximum I think 250 per year. So actually no, I shouldn't say that. Maximum of 500 in any 1 year, maximum 750 across the whole project. So I want to sort of finish out with some things to do, and the first thing, and I say this at every webinar, and I can't tell you how many times every time I get receipt dates, I have to call people up and say, you forgot to put this in or forgot to put that in that's in the funding opportunity announcement. So please read the funding opportunity announcements. No matter what program you're applying to, they all have sort of idiosyncratic requests in them. And so I wish they didn't, but they do. Ours is that you have to have an IP agreement, an IP plan as an attachment in ours, and it's telling you to put tables that clearly delineate what you're doing and what you're requesting the NIH to do through contracts. And that way it allows the reviewers to know what they should be reviewing and what they shouldn't be reviewing. It allows us to make sure that we can provide what you're asking for under NIH contracts and beforehand. Discuss your proposal with NIH staff. We're here to help. And I'm happy to talk to anybody about their program. I can't write your grant for you, but I can certainly give you the guidances that I can. And we can discuss where your program fits and tell you who the science officer is, because all of these grants are funded by the institute or center that your particular disease is under. So if it happens to be Parkinson's, it's going to be funded by NINDS. But if it's Alzheimer's, it's going to be funded by aging. If it's substance abuse, it's going to be funded by drug abuse, etc. So it's important that I know where you're going, where your primary indication is, and so that we can hook you up with the correct people in their institutes to garner the support. So if you score well, funding is streamlined. You have to stick to the page limit. It is not that large. I understand that, but don't try to circumvent it by making microprint and things like that, because CSR will just kick your application out. Make sure you put forth a solid scientific data premise with preliminary data that's in your research strategy section. Don't bury it in the vertebrate animal section with rigor and such, because you have to address rigor. Is it reproducible? Is it properly controlled, blinded, etc., powered for what you're talking about? Also address any obvious criticisms that you have for chemistry or biology. So if you have a structure alert in your molecule and you recognize it's there, you can say, I recognize it's there. It's going to be part of that exploratory effort that we have, is going to be determining whether we can get rid of that and maintain activity. That's fine. And it shows the reviewers that is your plan. But if you don't say that, then a lot of times the reviewers get stuck on, well, they have a flag in their molecule. They don't have any plans to get rid of it. This is not going to work. So you want to obviously try to address any obvious criticisms you have. In biology, if 14 other pharma have looked at this particular target or before, why are you going to be different? How are you going to succeed when others have failed? You need to address that question. As I mentioned, clearly indicate what's going to be done as part of the grant and what's expected to be done to the contractors. The grant requests a table, and I strongly suggest you put a table in, because it makes the reviewers and the staff here a whole lot easier to figure out what you're asking for. Include budget year sheets for every year of the grant. And every cycle, I have to call people and say, you forgot to get in the UH3 portion of your grant. Or you only put a grant in for that's going to the end of UG3 and not to the clinic. So, you don't have budget for the whole way in the program. So I call people up and say, you need to revise your grant application quickly. We've only been able to do that if you submit it early. With COVID going on, people have had a little more leeway, but that's eventually going to end, and the hard stop's going to come back again. As I said, preliminary data, make sure it's in your research strategy section. And not only your preliminary data discuss the rigor for, but if you're citing literature data, was that done rigorously? Discuss that briefly in your research strategy section, so people know. And what you're planning to do will be done rigorously, and it's going to address sex and age as variables, and make sure you're looking at that, because again, the reviewers are instructed to look to see whether you're doing that. Complete all your registrations at least 6 to 8 weeks in advance, and it's incredibly important. I've had this happen, unfortunately, to a couple of small businesses over the years, where they've waited to the last sort of possible month to get things going, and we're right up against it. And unfortunately, sometimes it just it takes longer than you expect. Sometimes you can do this in 2 weeks, but 6 to 8 weeks is where you should really plan to do. In most cases, it's very difficult to do it in shorter periods of time. Consider putting your application in early. I do look at applications as they come in the week before, and try to spot anything that might be an issue and why you still have time to edit it and change it back, or address the issue. So the earlier you can get it in, the more likely I have a chance to look at it and respond back to you if any issues are there. Do talk to your tech transfer offices about the IP and who's going to run it, and have that sort of letter of support. And as I mentioned before, if you're going to exceed the $500K in direct costs under the UG3, UH3 in any year of the grant, give me 6 to 8 weeks to go through the ARA process for that. So things not to do in the program: do not plan for this to be your sole funding source for your lab. It's milestone driven. It can end abruptly if science says we just hit a dead end. Don't under-resource your budget to avoid the limits. It's better to come in with an ARA over the limit than it is to find yourself under budgeted to be successful and you end up missing your milestones. Do not plan for BPN contracts to do disease biology. We just simply can't do that under our contracts, nor plan for us to do one-off in vitro ADME experiments, things, I need a SIP for this one compound or something like that. That you should do yourself. If you're going to do a whole program of in vitro ADME work, then we can do it. But single one-off experience is much better to have you just do that yourself. Do not code the application of clinical trial unless you are doing the clinical trial yourselves within your budget. So at your site, you're controlling it. Otherwise, it's not a clinical trial. And even if you do come in, it's probably a delayed onset trial because you don't really know what that's going to be like until we've had discussions with clinicians and others to finalize what we're thinking about. As I mentioned, BPN has a new program that is a sister program to it called BPN Biologics, very similarly laid out for antibodies, peptides, proteins, gene-based therapies, cell therapies, etc. The peptides are the cutoff for small molecules. I think it's the 6 MERS. How we do anything over that, 6 MER and up, we say can go to BPN Biologics because they have new contracts that are allowing a little more flexibility in manufacturing and so forth in this space than the small molecule space, which we covered previously. Chris Boshoff is the present contact for that program. It has a UG3, UH3, and a SBIR fast track. It has identical receipt dates to BPN. It is reviewed at the same special emphasis panel. First, we had one yesterday, and biologics went first, and chemistry went second. I don't know if it'll always be that way. But I think it's just based on reviewers' availability. But the reality is that's how it goes, and we look at those. They do allow up to a 2-year phase 2 in some of the biologic space. If you're in the pain area, these used to all come to BPN, but we have a sister program now through the Helping to End Addiction Long-Term Initiative, HEAL. This program is an RFA, so a set-aside set of money going to this activity. It's nearly identical to Blueprint Neurotherapeutics. It does require you to try to develop a biomarker for the clinic in terms of pharmacodynamic type biomarker that you could look at, target engagement biomarker that you could look at and see whether you can see something in the early stages of the program. Marianne Pellemounter is the program director for that. We have multiple receipt dates coming up in the next year, and they're listed here. So at this point, I'm going to open it up to questions. Those questions can really be about the application process. They can be about the review process, proposals and budgets, contracts, or intellectual property, or anything else outside of these 5 you could think about. So I'm happy to try to answer as many questions as we can. Thanks.
Thanks, Chuck. And let me remind everyone, please put all of the questions in the chat section that you should see below. Also, that this recording will be sent out along with the PowerPoint presentation in the near future. Okay, Chuck, we only have one question so far, so I can go ahead and read that to you.
Sure.
Is it okay if our company is a small business not currently affiliated with NIH?
I'm not exactly sure what that means. Small businesses. So first off, the answer is yes, no matter which way we go here, because small businesses can apply that are not SBIR eligible can apply to the UG3, UH3, no matter what. So if you're an SBIR eligible institution, you have to self-attest to that. Then you have to follow the SBIR rules to do that. And maybe Carolyn can post the SBIR website in our chat for people. But if you follow the SBIR rules, then you can certainly come to the SBIR, but you have to do that. If not, then you'd have to apply to the UG3, UH3. And so when does that come up? It comes up a lot in my program because a lot of times we're dealing with virtual companies. And so in order to meet the rules, we have to have 2/3 of the money staying into the company in phase 1 of the grant and over 50% in phase 2 is hard if you're a virtual company. So a lot of times those virtual companies come to the UG3, UH3 mechanism because they're not going to qualify for it under SBIR rules. They're still a small business. There's just not qualifying for SBIR rules in order to go off for an SBIR grant.
Okay. Next question. 1 second. It keeps getting stuck. Is there any interest in diseases that affect retina and photoreceptor degeneration?
Yes. So NEI is signed on to the FOA, and we've had projects in macular degeneration and other eye indications over the years. In fact, one of the projects in macular degeneration and Stargardt's disease is now in phase 3.
Next question. For lead optimization, are there NIH resources for L search for new chemical scaffolds?
AI search, artificial
Yeah.
intelligence.
Mhm.
We also have other resources available for new scaffolds. Yes, there's computational chemistry available through our Courier contract, and we have done virtual searches and scaffold hopping exercises, pharmacophore searches, and other things through the available chemical databases we have, like CDD, the relational database we talked about, has access to a whole bunch of commercial databases in there, so we could potentially search those. But if you're doing this yourself and developing it, it could still be done as part of the program. It just I'm not sure it would be lead optimization at that point. It'd probably be more in the exploratory or hit to lead phase.
All right, next question is, we wish to validate a plasma CSF biomarker for TBI patients. Is this the right grant to apply?
Now, if you want to develop a biomarker, there are biomarker funding opportunity announcements available through NINDS for doing what you're asking for. Again, I can ask Carolyn if she can potentially find a link to Carol's site and perhaps post that as well.
All right. Just to confirm, for the proposed budget, we don't include any of the proposed contract testing.
For your proposal, if you're the contracts that are NIH contracts, you do not put in your budget. If you have separate contractors that you are contracting yourself directly on your grant budget, then you could put that on there. But if you're utilizing the contracts I was talking about from the NIH side, then they're not part of your budget.
Next question. What are the pros and cons of the BPN versus SBIR mechanism?
Okay, they're both BPN. You're asking what's the difference in the UG3, UH3, and the U44 mechanism. So if you're an SBIR eligible organization, the advantages are that it's a separate pool of money and that you can charge a fee for the grant stuff. The contract stuff is all paid through non-SBIR money. So it's just potentially a different pool of money. The reality is the biggest advantage is that you can charge a fee, a profit that you could not do in the UG3, UH3.
All right, next question. Do any of your CROs provide assistance with planning preclinical tox studies?
Our CROs and our consultants are the ones that really help you with the planning stages of those tox studies. So we have tox consultants from that work with our teams and will help design exactly the kind of studies you need, everything from how many animals to what tissues to look at, depending on, again, that's where I say that, having that clinician involved to say, it may be an unusual tissue that you need to be including in your tox studies and looking at for this particular indication or this particular class of drugs.
Okay, next question is, we make glycopeptides,
7 to 27 MERS.
yes.
Can these be small molecules? Those would be biologics, simply because of their size, and our MedChem contract couldn't do that, so we would have to go through the biologics contract and sort of farm this out to a specialized organization to do that kind of work.
All right. Great presentation. Is it a sample application available for guidance?
To date, no one has put their successful application up for other people to look at. There are, I mean, you can look on our website and see who the present grantees are, and you could potentially reach out to them and see whether they would share their application. Obviously, the NIH can't share someone else's application. I tried to put in the instructions for the grant funding opportunity, the things I've seen in successful applications. That's why I asked for, make sure you put a screening funnel in, make sure you put a table that clearly delineates what you're doing, what the contracts are doing. You want to lay out your project from start all the way to the clinic, which is why I have those milestones up there, so that allows people to sort of say, here's the structure of how I want to put my grant organization. I need to go through these steps depending on where I'm coming through. So I try to give as much information as I can to help you lay out the application, but presently there is no sample application.
All right, next question. For lead optimization, are there NIH resources for SAR based on structural data, example, for a target enzyme?
So our Curia contract, which does medicinal chemistry, includes 3 components. It has synthesis, it has in vitro ADME, and it has computational chemistry. So the computational chemistry group can help you with the SAR-based structural data analysis.
Okay, someone asked, could you please advise on the resubmitting process?
Okay, so you get, so if you submit once and you're submitting a second time, it's called an A1 application. You can address the prior reviewers' comments as long as you're within the time frame allowed. Leave your allowed 1 or 2 pages to do that. You have to look at the instructions in the grant that shows, here's what I've changed, and you can highlight that in the grant application to say, reviewers' comments were this, and I did this to address those. And it's that simple. You put it back in again. I would strongly suggest after the summary statement comes out, if you plan on resubmitting, you have a conversation with the NIH staff, and we can go over your summary statement together and see whether, from my experience, or others' experience, whether they can help interpret what you're seeing there. There was no discussion if it wasn't discussed. So you'll have the same summary statement I have. If it was scored, then there was a discussion. And my sense of what they meant probably would be stronger than if I'm just reading it and it wasn't discussed. But the reality is I can't tell you exactly what reviewers said, but I can at least guide you on what I think are the most salient points to address.
Okay, next question. Thank you. I missed the opening part. Are BPN, SBIR, STTR grants considered fast track application, or is this a possibility of applying to phase 1?
So there is no STTR version. There's just SBIR version fast track. So there's no just phase 1. As I mentioned early on, all applicants are coming in, and the designs for wherever you come in to go all the way from where you started to the clinic. And so we'll have a fast track or a phased mechanism, but either way, trying to go all the way.
Okay, next question. How do I find the person at the NEI to discuss a small molecule to arrest rye photoreceptor degeneration?
So the easiest way to do that is to go to the funding opportunity announcement and look into who the scientific resources or contacts are.
Yeah,
Just,
sorry about that. I see Carolyn responded to her with the information.
yeah, but if you come back, I actually had it, and these slides will be available to you. But here are the contacts that are in there, and in the funding opportunity announcement, it will tell you. So here are the 3 contacts at NEI, and I think Dr. Lee does the SBIRs, and Tom and Niraj do the non-SBIRs mainly.
Okay, next question is, what is the current success rate?
It is similar to other programs. I honestly don't know. And it varies obviously by cycle and so forth. But overall, I would say it's pretty similar to R01 success rate.
Okay, now
I matched myself up to sort of DD&S R01 study section to see how I'm matching.
okay, and the last question so far is SBIR is a type of BPN. So do companies that receive SBIR funding get access to BPN resources? How does it work?
No. So there's an SBIR version of the funding opportunity announcement for BPN that allows SBIR eligible organizations to apply to our funding opportunity announcement as an SBIR organization. That allows you to then, like I said, charge a fee. And as long as you can follow all the SBIR rules, that's great. And it gives you another opportunity to come in. I mean, alternatively, if you have all the resources, you don't need NIH contract resources, you don't need NIH expertise, then I always tell an SBIR organization like that, they should probably just go to the omnibus. The people that come to BPN are coming because they lack the expertise in drug discovery and development, and we can provide them the consultants, we can provide them the contracts that they may or may not know they need in order to get to IND. But there is a trade-off for that. It's a milestone-driven program. It's much more linear than you might choose to do it if you were doing this strictly in a biotech world where you could do everything in parallel at risk. We can't do that in this milestone program.
Okay, another question. How does the BPN program work if there's a red flag somewhere in the pre-development period? Can you go backward and start lead optimization or hit to lead again?
I guess it really depends on where exactly we're talking. If you were very early, let's say you hit it in dose range finding, then maybe. If you're doing it in GLP tox, probably not. You probably have to come back in as a new application because it's just the cost and the amount of money spent in there to go back and start over. At that point, you know, the BPI has already spent a lot of money, and they're not going to probably want to go back without having it re-reviewed.
All right, next question. Can US-based company work with overseas contract research organizations to execute work in the grant?
Under UG3, UH3, you can. Under SBIR, you cannot.
We have so far. Give it a couple more minutes.
Okay. Great set of questions. And if anybody's going to be at SFN next week, I'll be at the booth. You can come talk to me and talk more. As such, if you try to set up a phone call with me in the next couple of weeks, you will find I'm probably not available until we get back, and then the week of Thanksgiving is coming up, so that'll limit the availability a little bit there. All right, if there are no more questions, again, I will print out a PDF version of the slides that we presented today and give that to Conda to send to all the people who signed up for the webinar. And if you have any questions, I would say give us a call, go to the website. There's a lot of information on the website, including stuff I didn't get to today. I hope you can think about our program when you looking to apply. Thanks a lot. Bye now.